next session without taking much of your time. So I welcome Mr. Shivaji Mante, Assistant Manager, Fluid Application Scientist with Premas Life Sciences. He's a person with 13 years of field experience of next generation sequencing, single cell sequencing and agricultural biotechnology. I welcome you, Mr. Shivaji Mante. On a lighter note, to tell about him, when we got Illumina machine in our hospital, the day one to give the, they give a proper training when we buy the machine. So one of his colleagues, Mr. Anil, has come and taught us till sample preparation. And the next day he chipped in, and my HOD told, be aware of this girl. So he actually literally held my hand, and he was even abusing me on a lighter note and taught me. So I welcome you, sir. The stage is all yours.
Because he understood you learned more.
No, if you are not giving you certificate. मैंने बताया नहीं देखूँगा.
The certificate is there.
Okay, thank you so much for the introduction. Yes. So I know there are lots of learning going on since morning, and we're learning a lot about all the sequencing platform over here. So I'm going to talk about Illumina next generation sequencing platform and how Illumina can help microbiologists doing their research work or doing their project. So talking about Illumina is world's leader in next generation sequencing. So Illumina manufacture, develop and market innovative next generation sequencing technology platforms and microarray technology. It has its headquarters in San Diego, US, and when you talk about next generation sequencing data, out of entire sequencing data generated, Illumina holds 90% share globally, and we have almost 350 plus platforms installed in India. So I will start with Illumina sequencing overview. So this is the basic of Illumina sequencing technology, how Illumina sequencing technology works, and it is common across all sequencing platforms, like when you talk about the smallest or the highest throughput platform, the Illumina sequencing overview is common for all sequencing platforms. So these are the four basic steps of Illumina sequencing technology. The first part, library preparation. Second part, cluster generation. Third one is sequencing, and the fourth one is data analysis. We'll see all these four steps one by one. So the first step, library preparation. So the library preparation is something which happens outside the sequencing system, right? So what you do basically, you take your nucleic acid sample and you attach known nucleic acid sequences to your unknown nucleic acid sequences. So in the simplest form, you add the known sequences to the unknown part, and then the unknown sequences are being read by the sequencing platform. So how basically we do that? So this will be, so you will start with nucleic acid. So if it is RNA, you need to convert that into a complementary DNA. If it is DNA, you start with the DNA for the library preparation. So Illumina sequencing technology is a short read sequencing technology. So you need to fragment your DNA into shorter fragments, and you do that by two methods. One is enzymatic or physical method. You use ultrasonic filters such as CoVaris. Okay? So you fragment your DNA into shorter fragments, then you ligate the adapters to the fragmented DNA, and then you amplify the entire library structure, and finally you perform a cleanup, and then you get your library which is ready to load onto the sequencing platform. Okay, so when we talk about the library, your library, ideal Illumina library structure looks like this. Okay, so when you see this P5 and P7 adapter sequences at both ends of your library, so these are the adapter sequences which helps in the cluster generation. Okay, so what are the clusters? I will talk in the next slides. Next to that, is your index 2 and index 1 sequences. So this index 1 and index 2 sequences are the nucleotide sequences which provides molecular identity to your each sample. Okay, so when you perform a PCR or a qPCR, so you put your each sample in a different well, right? You don't mix your two samples in a single well, right? So just to provide identity, you give a physical separation, right? But when you talk about Illumina next generation sequencing, you pull all samples together, load it into a single well, we call it as a library loading position, and then these all libraries are loaded onto the flow cell. Okay, so unlike Oxford Nanopore, we everything is loaded onto the flow cell by system itself, and then your cluster generation happens. Okay. So this P5 and P7 adapter sequences are held for cluster generation. Index 1, index 2 provides the molecular identification, which helps in pooling n number of samples together, as well as once you generate the sequencing data, in order to differentiate data between two samples, this molecular barcodes or indexes help. So the process is called as demultiplexing. Okay. Then next you have RD1SP primer binding site and RD2SP primer binding site. This RD1SP primer binding site helps in the sequencing in the forward orientation. Okay. So Illumina sequencing can do sequencing in both the direction. So we call it as a paired sequencing. I will talk about that again. And next is your read-to sequencing primer, which helps binding of read-to primer and performing sequencing in the reverse orientation. Okay. So this is how an ideal library structure looks like. And in between these two structures, you have your DNA insert which you want to sequence. Okay. So after that, the next step is cluster generation. So this is the first step which happens onto the sequencing platform, where you basically amplify your single stranded library molecule into multiple copies, approximately 1000 copies. So what you do basically, you load your single stranded library. So when you prepare your library is double stranded. So basically you provide a high alkaline environment, you denature it into a single strand so that this single strand molecule will go and bind onto the flow cell. If it is double stranded, it will get washed off, it will not go and bind onto the flow cell. Okay. So when this single stranded DNA molecule is amplified into 1000 copies, this 1000 copies altogether called as a cluster. If you look at this image, each bright spot represents one cluster. Okay, so this is the image from a MySeq sequencing run or a random flow cell sequencing run, and each dot here represents one cluster. So, like since morning you have heard about flow cell, right? Whether it's Oxford Nanopore or it's Illumina. So the entire chemistry happens onto the flow cell. Okay. So in case of Illumina, this flow cell is made up of glass surface or a sandwich of glass, in between which the reagents flow, and as the reagents flow and the entire chemistry happens, this is called as a flow cell. So in between the sandwich of two glass surface, the reagents flow. So this glass surface has a specialty. Okay. So if you look at this structure, which is a magnified version of this from a flow cell, so it has two types of oligos which are complementary to your P5 and P7 adapter regions. Okay. So using this lawn of oligo, which is randomly coded onto this flow cell, your library molecule will come and bind onto the flow cell, and the next process of amplification will happen. So when you talk about different sequencing platforms, the flow cells used in platforms are of two types. The sequencing system may have either random flow cell or pattern flow cell. So in a simpler term, the random flow cell platforms have lawn of oligos which is randomly coated onto the glass surface. It is not having any fixed shape or size or dimension. It is randomly coated. Hence the clusters generated are of randomly placed and of random size. Okay. So then when you talk about this random flow cell, it is used in ISEQ 2500, MySeq, NextSeq 550 and MiniSeq. Onto this random flow cell, you get comparatively lower duplication rates. On the right hand side, you can see a flow cell, we call it as a pattern flow cell. As the name suggests, it has a particular pattern. So when we say pattern, onto this glass surface, the nanovels are coated, and the lawn of oligo which is randomly placed here is only at the bottom of the nanovel. Okay, even if it is not on the top of the nanovel, it is just at the bottom of the nanovel. So because of this pattern flow cell, so this pattern flow cell is used in ISEQ series, NovaSeq, ISEQ 100 and NextSeq 1000, 2000. So because of this pattern flow cell, what happened? The clusters is of defined size and space. So like if you look at these structure over here, you can see like each wells are at particular distance from each other, and even if you can see that, so as the clusters will generate inside these nanovels, they will get a defined size and shape, right? So it increases the cluster density that you get onto the sequencing platform, and the imaging process becomes simplified as compared to the random flow cell. Okay. So next process is how the cluster generation happens. Okay. So the cluster generation happens in a random flow cell is a traditional cluster generation. What happens in a pattern flow cell is a exclusion amplification cluster generation. Technically the process is same. The only thing is in a random flow cell it happens in a random way onto the flow cell. In a pattern flow cell it happens inside the nanowave. Okay. So when you load your single stranded library molecule onto the flow cell, it binds to the lawn of oligo, and then the 3' extension happen using polymerase, and a new strand will be synthesized. Once you synthesize a new strand, the original template molecule is cleaved off using a chemistry step. Now you have a newly synthesized strand. After that, what happened? This single strand linearized molecule will flip down to the adjacent oligo. So as you know, there are B5 and B7 adapters on the end of your library molecule. So, and the lawn of oligo has two kind of oligos which are complementary to your B5 and B7. So it will flip over and bind to the adjacent oligo. Again, the polymerase will extend the strand and a new strand will be synthesized. If you look at this structure, it forms a bridge-like structure. Hence, this process is called as bridge amplification. After that, again this double-stranded bridge will linearize and they will form two separate strands. Again, they will flip down, form a bridge-like structure. Again, second strand will be synthesized, and this process will keep on happening until 1000 copies of such molecules are generated. Again, your double-stranded bridges are linearized. Now, on the flow cell, you have strands which are in forward orientation and reverse orientation. So the very first point that you want to do is, you want to do sequencing for the forward strand. So what you do, you cleaved off the reverse strand, leaving behind only forward strand onto the flow cell. So after that, read-one sequencing primer will come and it will bind onto the library molecule at RD1SP primer binding site. So if you remember, I told you about RD1SP and RD2SP primer binding site. So the RD1SP primer binding site is used for sequencing in the forward orientation. So here, after that, once your read-one sequencing primer comes and binds to the library molecule, your actual sequencing will start. So the sequencing that happens onto the Illumina sequencing platform is sequencing by synthesis. So I haven't elaborated that here. So basically we have three different chemistries. One is 4-channel chemistry, one is 2-channel chemistry, and one is single-channel chemistry. So today I'm just going to talk about 4-channel chemistry. In short, the number of fluorescent labeled dyes are used. So in 4-channel chemistry, 4 different colored fluorescently labeled dyes are used. In 2-channel chemistry, only 2 different colored fluorescent dyes are used. And in a single channel, the chemistry happens using a single fluorescently labeled dye. So before we move to the actual sequencing, so how the sequencing by synthesis happens, I would like to talk about the Illumina sequencing platform. So if you look at this slide over here, the smallest in the chart is IC100, which can generate 1.2 gigabases of the data, and onto the flow cell you can generate 4 million reads. Next to that we have Mini-C. Mini-C can generate up to 7.5 gigabases of the data output, and it can generate up to 25 million reads onto the flow cell. Next to that, you must have seen this platform. This is Illumina IC. Illumina IC can generate up to 15 gigabases of the data output, and it can generate 25 million reads onto the flow cell, and it can go up to 600 base pair length for the sequencing read. And next to that we have Nexit 550. So Nexit 550 can generate 120 gigabases of the data output, and it can generate 400 million reads onto the flow cell. The maximum read length can be 300 base pair. Nexit 1000, 2000 are the recent launch from Illumina. Nexit 1000 can generate 120 gigabases of the data output, and Nexit 2000 can generate 360 gigabases of the data output. So if you look at this image, physically both the instruments are same. The Nexit 1000 has capability to run only P1 and P2 type of flow cell, and Nexit 2000 can run P1, P2 and P3, all 3 types of flow cell. So a software upgrade can convert Nexit 1000 to Nexit 2000. And then we have IC 4000 and IC Next. So this series has been obsolete now. We no longer sell this instrument. And on the top of this chart is we have NovaSeq 6000. So as the name suggests, this NovaSeq 6000 can generate 6000 gigabases of the data output. So it has 2 sites to set up the sequencing run. Each site can generate 3000 gigabases of the data output with 10 billion reads onto the flow cell. So now we look at the 4-channel sequencing by synthesis chemistry. So this chemistry is used by HiSeq and MiSeq. sequencing platform. So as the name suggests, in this chemistry, each nucleotide is labeled with a different colored fluorophore. Okay, so 4 different colored fluorophores are used to identify all 4 bases. So G is labeled with blue fluorophore, T is labeled with green fluorophore, A is labeled with yellow fluorophore, and C is labeled with red fluorophore. So as the name suggests, there are 4 different fluorophores in each sequencing cycle. System needs to capture 4 different images to identify clusters with 4 different bases or 4 different colors. So what happened during sequencing? So if you look at this is your template molecule to which regional sequencing primer has bind. So inside the flow cell, these 4 fluorescently labeled nucleotides will be released, and based on complementary nature, one fluorescently labeled nucleotide will come and bind here. So this fluorescently labeled nucleotide has 2 distinct features. The very first one is it has a fluorescently labeled dye which is specific to that particular base. Second thing is, if you look at this structure over here, so this structure is called as reversible terminator. So what this reversible terminator does, when this fluorescently labeled nucleotide come and bind over here, it doesn't leave any free end to this nucleotide, in result of which a second nucleotide cannot come and bind here. Okay, so once this nucleotide comes and bind to the template molecule, the fluorescent label dye will be excited using light source, and a light wavelength of which is specific to this fluorophore will be emitted, and that will be captured by the camera. So if you look at this, so as if you have a single template molecule and you try to capture the wavelength emitted by camera, that is not possible. That's why we amplify the single library molecule into 1000 copies so that the signal also amplifies. So after that, once the intensities are captured by the camera, this reversible terminator is removed using a chemistry step. So now this nucleotide has a free end. So again fluorescently labeled. So this entire process from release of fluorophore till removal of reversible terminator is called as single cycle. And a single cycle, a single base is called. After that, again in the next cycle, fluorescently labeled nucleotides will be released inside the flow cell, and this process will keep on happening for the read length that you have mentioned. Okay. Sorry. Okay. So I was talking about parent sequencing. So Illumina has a patented technology, we call it as a parent sequencing. So in a simplified term, the sequencing platform is able to sequence your library molecule in both the orientation, that is forward and reverse orientation. This process is called as parent sequencing. So the parent sequencing helps in easy assembly of your genome. So what happened during parent sequencing? So now you are done with your sequencing in forward orientation. So the synthesized strand will be then peeled off, and again the linearized molecule will be flipped down. Again using 3' extension, the new strand will be synthesized. Again this double stranded DNA will be linearized. So at this point your forward strand is cleared off using a chemistry step, leaving behind only reverse strand because you want to do sequencing in the reverse orientation. So after that, read to sequencing primer will come and bind to RD2SP primer binding site, and the sequencing by synthesis will happen in the same way as it has happened for the forward strand. So now you are done with sequencing of both the orientation, forward strand and reverse strand. Now you wanted to do sequencing for your index bases, right? So unless and until you don't do sequencing for your indexes, you will not be able to distinguish data between two samples. So what you will be doing after that, you will be doing sequencing for index reads. So as you have seen in the library structure, you have two indexes, index 1 and index 2. So what happens, the very first time the sequencing will happen in the forward orientation. After that, the index, the I7 sequencing primer or in index 1, it will bind to the index, it will bind to the primer binding site, and then sequencing will happen for the index 1. The index 1 is also called as I7 index. After that, this single stranded molecule will flip down, and using P5 grafted primers which are there onto the flow cell, the I5 index will be sequenced. Okay. and after that the parent turnaround will happen, and then sequencing will happen for the reverse strand. Okay, so this is how the workflow happens for the entire reads, forward, index 1, index 2, and the reverse strand. So once your sequencing is done, the last step in the workflow is data analysis. So the data analysis happens at 3 different stages. Okay, the very first is primary data analysis that happens using control software and real-time analysis software in the background. So what this control software does, it captures the images and the intensities that are being excited and captured by the camera, and you get the base calls. Okay, so base call is the raw output format of your sequencing data. After that, the next step is analysis software. We call it as a secondary analysis. So in the secondary analysis, it happens using base-based sequencer, which is Illumina's cloud-based solution, or it also happens onto the system using software such as local run manager. So what this base-based or local run manager does, it will align your sequencing data to the reference genome, and you get to know about the variant detection. Then using tertiary software or visualization software, so it can be base-based or any third-party app, you get to know about annotation, filtering, and reporting of your sequencing data. So once your sequencing done, the most important thing that you get to know about quality scoring. Okay. So when you talk about quality scoring, so the quality scoring is nothing but the estimate the probability of error in base calling, and it gives you the quality model and it gives you the quality score. So what this quality score tells, the quality score ranges from Q10 to Q40. So when we say Q10 quality data, it means when 10 bases are called, there is probability of error in one base call. So this is 90% base call accuracy, we call it as a Q10 score. When we say Q20 score, when 100 bases are called, there is probability of error in one base calling. This is 99% base call accuracy, we call it as Q20. When we say Q30, when 1000 bases are called, there is probability of error in one base calling. We call it as Q30, and it is 99.9% base call accuracy. And when we say Q40, when 10,000 bases are called, there is probability of error in one base calling, and base call accuracy is 99.99%. So the data that we specified, it is in the range of Q30 to Q40. So the base call accuracy is quite high. It is ranging from 99.9% to 99.99%. So before we move to the next part of the presentation, I would like to play a short video, and then I will proceed to the next part of the presentation.
Preparation begins with extracted and purified DNA. The first step in Nextera sample preparation is tagmentation. During tagmentation, transposons simultaneously fragment and tag the input DNA with adapters. Once the adapters have been ligated, reduced cycle amplification adds additional motifs, such as the sequencing primer binding sites, indices, and regions that are complementary to the flow cell oligos. Clustering is a process wherein each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, 4 fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determine the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' end of the template is deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Index 2 read product is washed off at the completion of this step. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends blocked. The original forward strand is cleaved off and washed away, leaving the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is washed away. This entire process generates billions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments.
So I hope that video explains the entire process very well. So now we'll move to the next part, like how the next generation sequencing can help microbiologists or microbiology experiments. So World Health Organization says that the health of human population, the health of domestic animals, and the health of wildlife and the state of the ecosystem are intrinsically linked. And if you look at this diagram over here, so the antimicrobial or antimicrobial resistance, so this is a new challenge and it is being interlinked, and to understand this in a better way, genomics can help. Okay, so genomics can help in critical areas such as infectious diseases research and public health management. So next generation sequencing helps you to identify and characterize the pathogens which are challenging. You can help understand their mechanism. You can the genomics will help to understand the transmission between organisms, as we have seen in case of SARS-CoV-2. It will help to monitor the emerging of variants and mutants. So, which is again we have seen in case of SARS-CoV-2. It helps in the epidemiology and surveillance of the pathogens, and the next generation sequencing also helps to develop therapeutics to fight against them. So, genomics will help in different domains, such as if you want to understand the host genetics, host immunity, virulence and pathogenicity, antimicrobial resistance, drugs and vaccine development. If you want to identify the pathogen evolution or the pathogen origin tracing and spread. So genomics helps you to understand all these things. So there are different approaches, those are used to identify these things. So the whole genome sequencing, metagenomics or metatranscriptomics, enrichment or amplicon are few of the approaches that are being used to identify these things. So in whole genome sequencing, basically you target a microbial isolate, you do the whole genome sequencing of the DNA extracted from the isolate, and you came to know about the complete microbial or plasmid genomes from a cultural isolate. So when you have a pure isolate, you can perform a whole genome sequencing. When you have a primary sample, when I say primary sample, it is not a purified sample, it can be a mixture of different species, and you can, when you are doing the entire DNA analysis from the primary sample, it is called as metagenomics, and when you are targeting the entire RNA analysis from a sample which can be a mixture of different species, it is called as metatranscriptomics. So in terms of SARS-CoV-2, basically you extract the RNA, human RNA basically, and you enrich the SARS-CoV-2 part and then you analyze that, right? So then next one is enrichment, where you the organism is known and you target the known part. Okay, so that is called as enrichment. Same goes with the amplicon, where you in that case you amplify the target region and then you analyze that using sequencing. So next is microbiology next-generation sequencing methods and workflows. So this is again a little more descriptive explanation of the previous slide. So depending upon the kind of target organism, whether it's known, suspected or unknown, you can decide the strategy to identify that sample. Okay. So when the target organism is known, you can make use of targeted DNA amplicon sequencing. If you want to study the RNA part, you can perform a targeted RNA sequencing. When you have a suspected organism, you can either perform a whole genome sequencing or whole transcriptome sequencing. You can also perform a custom DNA enrichment metagenomic sequencing or custom RNA metagenomic sequencing. Next is when the entire content is unknown, right? You don't know like what this sample has. So if you want to understand the entire content of your unknown sample, you can perform a shotgun metagenomics, or you can perform a metatranscriptomics for RNA part of your unknown sample. So these are the approaches that can be used. So next is culture-based whole genome sequencing. So this is a part where when you have a culture or isolate of your sample. So for bacterial cultures, you know the genome size and you can target the 100% of the organism. You can target, for example, the whole genome sequencing is ideally done at 30x coverage when the species is known or the sequencing is already done. If you are trying to do something novel, you can target 100x or 200x coverage. When you are wanting to study the virus part, so if your virus is RNA or DNA virus, or if it is DNA virus, if it is single-stranded or double-stranded virus, depending upon that you can decide the approach to perform the sequencing. So this slide talks about viral isolates, so how you can target different approaches to study different viruses. For example, if your virus is a RNA virus, the very first thing that you need to do is you need to convert your RNA into a complementary DNA, right? So the first thing is you will perform a complementary DNA, you will perform a cDNA synthesis using reverse transcriptase. Then the single-stranded DNA will be converted into a double-stranded DNA, and then you can prepare a library using Illumina DNA prep, and then you can perform the sequencing on any of the MySeq or NextSeq platform, and then you will be doing the data analysis. If you are So if you can also make use of closing RNA sample preparation kit, which have all the reagents required to perform cDNA synthesis and everything, and then you can perform the sequencing. If your virus is a single-stranded DNA virus, then you need to convert your single-stranded DNA virus into double-stranded DNA, and then you can prepare library using Illumina DNA prep, and the sequencing can be done on any of the sequencing platform, and then you will be doing data analysis. If it is double-stranded DNA virus, it is quite straightforward. You can directly prepare library using Illumina DNA prep. You can do the sequencing on any of the Illumina sequencing platform, and then you can do the data analysis. So Illumina DNA prep is a ideal kit for performing all of these applications. So because this kit works with broad range of DNA input, and it has an integrated protocol, you can start with blood, saliva, dried blood spot, or any of the microbial colony as the input. Then it gives you high uniformity and coverage. It gives you a consistent insert size because of this built-in transposons, and the workflow is quite flexible. In 3-4 hours of time, your library will be ready. It works with the DNA input range starting from as less as 1 nanogram till 500 nanogram, and it gives you a consistent insert size. So these are the input material that can be used with Illumina DNA prep. You can start with genomic DNA, you can start with blood or blood punch card, saliva or a bacterial colony. So these are few of the methods that will be you will be starting with. The very first part will be your DNA extraction, DNA quantification. Then you will be perform DNA sharing using weeding transposon. Then you will be doing adapter ligation, index tagging, and normalization. So this is the standard mechanical method. When you use standard enzymatic method, you will be doing the same thing. But here in Illumina DNA prep, so the DNA extraction, fragmentation, and index tagging and normalization takes very less time because you need not to normalize your input DNA, right? So because there are few library preparation kits which ask a specific amount of input DNA, here you can give a range starting from 1 nanogram till 500 nanogram. Hence, no normalization is required. You are saving onto the unique reagents. And then the fragmentation. So you have heard of fragmentation, right? So fragmentation is fragmentation plus adapter ligation. So the two processes together called as fragmentation. So in a single step, you are doing two steps. So now we'll see in detail about different approaches for the metagenomics. So metagenomics is nothing but you want to understand the species content or the diversity that a primary sample has. So when I say metagenomics, it is not for a pure sample, it is for the a primary sample which may contain n number of species or different species inside it. So it can be your sample from water, or it can be a soil sample, or it can be a so we call this as an environmental DNA which contains DNA from different species. So there are two approaches when you talk about metagenomics. The very first approach is targeted metagenomics. So when you want to study a particular species, right? You can target 16S RNA for bacteria and archaea. You can target 18S or ITS for eukaryotes such as fungi. So this gives you phylogenetic marker to define which lineages are present in your sample and which species are in abundance. Then when you say short-term metagenomics, you get to know about the entire gene content, metabolic pathways for all the species which are present in the sample. You get to know about relative abundance of microbial taxa, and you come to know about total DNA and RNA content. The only difference is when you talk about targeted metagenomics, the sequencing depth that is required is very less. We call it as a shallow sequencing. When you talk about short-term metagenomics, you need to sequence your primary sample at very high depth of coverage. It can be starting from 5 million to 50 million reads. So the metagenomics is an application of high throughput genomics techniques to study the communities of microbial organisms which are directly in their natural environment. So if you look at these two samples, it can be either from your soil or it can be water sample from either lake or sea or different location. So or even if it can be the same sample from the same location with different treatment, time point or different season. So what you do, you collect the sample, you extract the DNA or RNA, you prepare library. So if you are doing a targeted sequencing, you can do a 16S or IDS, or you can target the entire part, and then you are doing the sequencing onto the sequencing platform, and afterwards you come to know about the relative abundance in that sample. And you also get a comparative analysis if you are collecting samples at different time point or different season. So these are the two approaches. So in the targeted sequencing, you can also 12S, 16S, CO1, 18S or ITS sequencing. You get clade specific information. When you are doing short-term metagenomics, you get to know about the taxa, relative abundance and function of the entire species which are present, which may be bacteria, archaea, fungi, virus or proteome. So there are different demonstrated protocols available onto the Illumina sequencing website. We have a demonstrated protocol for 16S metagenomic study. We also have take note which talks about 16S metagenomics onto the MySeq sequencing platform. We have a fungal metagenomic sequencing demonstrated protocol and take note for the same. So this is a slide which talks about a comparative study done using targeted and shotgun metagenomics. So if you look at this both graph, so on the left hand side, this B and D are the charts from the shotgun metagenomics, where the deep sequencing is done with 50 million reads per sample, and on the right hand side, it is the shallow sequencing done with 500,000 reads per sample. And if you look at both the charts, which are quite comparative, right? So the shotgun metagenomics gives direct access to the genetic content of the entire community. So you can target different samples from different region, and you will get to know about the entire content of that sample. It talks of, so the shotgun metagenomics helps in the reconstruction of the genomes in the objective, and it also talks about the annotation of the entire community to look for the specific genes of interest, gene function, and taxonomic neighbors. So the shotgun metagenomics provides a comprehensive analysis of all the genes and organisms present. It talks about the bacterial diversity, and it detects the evidences of the microbes in the various environment. It studies the unculturable microorganisms that are otherwise difficult or impossible to analyze. And even if you can think of, like, if you want to isolate and culture all the microorganisms which are present in your sample, so it will be very tedious and time consuming, right? So here you need not to culture anything, you just need to extract DNA from your primary sample, and you can perform the sequencing. So the NGS based metagenomic sequencing can detect very low abundance members of the microbial community that may be missed or too expensive to identify using other methods. So this is the workflow. So the very first thing is you will be performing nucleic acid extraction from your sample. Then you will be preparing library using different library preparation kits such as Nextera, Nextera or Proceed DNA PCR. Then you will be performing sequencing onto a sequencing platform, and then finally you will be doing analysis. So again, these are few of the advantages of the shotgun metagenomics. The very first is it is culture independent. You need not to perform any culturing. It is hypothesis free testing. You can do multi-kingdom analysis. It gives you relative evidence of microbial taxa, pathogen detection, and again with newer advancement in the library preparation kit with unique dual indexing, which will address the index hopping issues, and you can perform metagenotyping. It will also gives you idea about the potential enzymatic function in the microbiome, and it will also give you idea about the metabolic pathways. So now next we will see about the infectious diseases. So now we have seen, like, the zoonotic transmission of infectious diseases is a serious threat, and we have, all of us, we have actually faced that in last 3 years. So there are different animal hosts, and we have seen transmission to humans, like SARS, starting from SARS 2003 till SARS-CoV-2. So we have seen all of these different zoonotic viruses, and in order to identify like different zoonotic viruses, there are different approaches. So in this slide, I will be talking about Illumina's COVID-6 portfolio. So how we have performed the SARS-CoV-2 sequencing using different approaches. So in the Amplicon approaches, we have used COVID-6 library preparation kit, which can perform detection, diagnose, and it can also help in evolution monitoring and screening of your SARS-CoV-2. We have an enrichment panel, we call it as a respiratory virus oligo panel. It can help in detection of co-infection along with the SARS-CoV-2. It can identify different respiratory viruses. It can also help in performing the evolution monitoring of different respiratory viruses, and it can also be used for the screening of different respiratory viruses. You can also make use of shotgun metagenomics approach, which I have already explained in my previous slides, and you can also make use of respiratory pathogen identification panel, which helps in identification of co-infection along with respiratory viruses, bacteria, fungi, viruses, and antibiotic resistance. It can also identify SARS-CoV-2 and flu viruses. It can also be used in the screening part. So this is the respiratory virus oligo panel, which is designed to detect and characterize respiratory virus co-infections. It is an enrichment-based approach which can identify all major respiratory viruses. So this is the list of different 40 plus viruses which can be sequenced using respiratory virus oligo panel. It has 7,800 probes which are 80 plus. So next is about drug resistance. So antibiotic resistance is another trait that we have, like with new pathogens having different resistance to different antibiotics. So we have seen that 27 million people get unnecessary antibiotics, and like 30 million are who don't need antibiotic out of that. So we have a respiratory pathogen ID library preparation kit which has a workflow which can detect the respiratory pathogen along with antimicrobial resistance. So it can do a detection of more than 280 pathogens, 1200 antimicrobial resistance alleles which are associated with the 16 antibiotic classes. It can also give you the full genome information about SARS-CoV-2, influenza A, B, A and B, and it also performs information about epidemiology and understanding the transmission of different viruses. So it also comes with the XXVI platform from IDY which performs the analysis of your sequencing data. So this is again the different list of different bacteria, viruses, fungi that can be identified using RPIAP panel, and this is the list of antibiotics that can be targeted using RPIAP panel. We also have Illumina antimicrobial resistance panel, which specifically used for detection of genes associated with the antimicrobial resistance. It targets 478 genes using 815 amplicons, which is spread across 2 different tools. So it can identify 478 AMR genes. So I'll just skip this. I will just go to my last part. I will just skip few slides. So now I will just talk about MTB solutions in 2 slides, and I will conclude my presentation. So in order to identify MTB genomic, we have 2 solutions. So you can basically target 2 approaches. One is whole genome sequencing and the other one is targeted sequencing. So the whole genome sequencing can be done using XXVI or Illumina DNA prep. And in the targeted sequencing, we have duplex Mycobacterium TB panel from GenoScreen. So this duplex Mycobacterium TB kit provides, this is the innovative kit used for MTB identification and drug resistance prediction. So like with the current phase, like you can identify TB either using microscopy, culture-based method or rapid test, but you will get limited information, and which will be again time consuming. So in order to overcome all of these pains in these 3 methods, there is a duplex kit which identifies the TB strain and its antibiotic resistance to 15 antibiotics, which can be done in less than 48 hours without any culturing step. So you can also identify mixed infections, and it will give you a digital visualization of the results using cloud-based web application. So you can get resistance prediction of resistance to 15 antibiotics. You will also get to know about genotyping and oligotyping of MTB strain. You can identify 156 mycobacterial species. The turnaround time, as I told, is very less, and it is highly sensitive. It can characterize more than 98% of variants which are involved in MTB antibiotic resistance. So with this, I will just conclude. I know I am taking a lot of time, so I will just conclude this with this. And if you have any questions, please feel free to reach out to me on these details. And thank you for your patient hearing.
Are you participating in the exhibition talks?
Yes.
And you
We are there for the
next what was this material available?
Yes, it is that.
And I will be interested in
Sure.
With our felicitation, Dr. Smriti, please hand over him memento and certificate. That was a very elaborative one on Illumina. Yeah. I call upon our next speaker, Dr. Kamlesh Jangid sir, a PhD scholar on molecular taxonomy and quantum sensing from NCCS in 2005. He is currently with HiMedia Laboratories heading Genomics and Bioinformatics Division. He completed his postdoctoral training in soil microbial ecology from USA, and he has more than 60 articles which have been cited more than 2,300 times. He also serves as editor to a couple of international highly indexed journals. He is currently president of Verghese International Society for Microbial Systematics. He was awarded honorary academic advisor appointment by China Agricultural University, Beijing, and is a recipient of 2017 Albert Menzel Lifetime Achievement Award by Marcus, USA. We welcome you, sir.
And thank you, Karikarte sir, for inviting me. I promise I won't extend my talk because I know everybody is rushing home and we are already 3 hours behind the schedule. So I'm here today not as a representative of HiMedia, but as a person who's been doing sequencing for half of my life. So I'm going to share my passion with sequencing and bioinformatics, and that's what I'm here to do today. HiMedia as a company started in 1976, and I'm sure of everybody in microbiology knows about HiMedia because you guys use a lot of media products. But there are many aspects of HiMedia that we are not aware of, and one of them is molecular biology. And HiMedia has been at the forefront of the COVID times where the DNA extraction kits, RNA extraction kits, real-time PCRs has been, something that have put the company ahead. So some of the COVID chain of products that HiMedia has is the coronavirus multiplex probe PCR kits. These are all CIVD approved. We have viral RNA purification systems, magnetic beads, extraction-free RT-PCR methods. These are dry swabs where you don't need to extract RNA. You can directly take the sample and do RT-PCR on that. We have bead-based methods. We have InstaQ real-time machines. So you have a complete portfolio of solutions that we are able to offer. Now, post-COVID, COVID is still there, I know, but we are not really worried about it anymore because there are many more diseases that are coming up. So the biggest bottleneck that was during COVID was actually the extraction of RNA because that was the step that was taking longest time. So the highlight of clinical diagnostics that's going to come is that you need to have a single extraction method regardless of the virus type. Whether it's an RNA or a DNA virus type, you have to have a single extraction method so you don't have to worry about using a different protocol for a different type of virus. So that's what HiMedia has pioneered in. We have a single extraction kit with individual cartridge. This is the NV582 variants. These are available in multiple platforms. You can buy them, and that's mostly about what our extraction kits can do. And we have a complete portfolio of real-time PCRs from the medicine perspective. We started with HPV, influenza, COVID, flu, malaria, dengue, serotyping and dengue chikungunya multiplex, and we also have HLA-B27. All of these are CIVD approved kits, and we have 3 in development that are already in the market, research use only, HCV, HPV and HIV. And we have just yesterday released the monkeypox kit for real-time PCR. And the reason I am highlighting all of these kits is because since the time I have joined this company, we have actually given it a bioinformatics backbone to ensure that the kits perform to their expectation and there is no cross-reactivity, there is 100% specificity to the target gene that we are looking to target at. And that's the reason why I wanted to put this. This is the monkeypox virus kit that we have come up. Just yesterday it was released. It's an RUO based kit, and as you can see, it can identify the monkeypox target as well as the parapox family, the entire family. So you have a distinguishing feature along with an internal control. And the AMR panel is also backed by hardcore bioinformatics that we have carried out, and all of these panels are CIVD. So these are very heavily used in medical sector, especially for detection of UTI strains or isolates that you have, or you can directly isolate DNA from the sample and do your panel assays. And that's about the reason why we are now diving deeper to rise higher into the sequencing and bioinformatics sector. And just to quickly tell you the history of sequencing, 1977, the year I was born, is when sequencing was born. And the yellow highlight is the one since the time I am doing sequencing in this field. So half of my life is sequencing. I started with radioactivity based gel method of sequencing, then I moved to single capillary. From there we moved to 8 capillary, 96 capillary, and then I pioneered pyrosequencing at University of Georgia, and then I moved to India to start Roche, took over 454, and then Iron Torrent came, then Illumina came, and today Oxford Nanopore and future 10x Genomics, all of these companies I have had the experience of working. So with this is the state-of-the-art sequencing lab that we have at HiMedia. As you can see, we have 2 Illumina machines, 1 ABI Sanger sequencer. We have a in-house Powerhouse bioinformatics cluster that we use to perform the entire bioinformatics. And we are NABL compliant, we have ISO norms, and the facility is fully functional, well equipped, and we offer a 360-degree genomic solution from sample to results. You can send the sample at any state, whether you have a microorganism, a tissue sample, a soil extract, or DNA, or even a library that is prepared at your end. Just because you don't have a sequencer, you don't have to hold your sample. You can send the sample to us and we'll take it from there. So it doesn't matter at what stage you want to submit the sample. We do the entire sequencing as well as the bioinformatics. These are the 4 portfolios that we cover: Sanger sequencing, NGS, genotyping, and bioinformatics. Sanger, we offer single double pass microbial identification ready to load. I'm aware that many of you are microbiologists and you have a need to identify organisms where the aircrafts are actually flying and they cause problems. So we are able to identify them. Our aviation specialists are working with bird specialists to reduce the time or change the timings of flights. So we are working with entomologists, we are working with serpentologists to identify the snake species in rural area. So a lot of things are able to be done. Single pass is 700 base pairs, single read for every base. 1200 base pair double pass is we read from both the ends. This is typically for pharma requirements or medical requirements. 96-well, you can do a 96-well, and you can identify any type of organism, as I said. NGS, we are able to offer whole genome sequencing. Whole genome sequencing, whole exome sequencing, microbiome, metagenome, and we have 2 different platforms. So regardless of the sample, some time ago I was overhearing there was a gentleman who was asking whether we can multiplex different types of samples. Like, let's say you have 2 organisms to identify. One is a bacterium, one is a tissue sample. We can multiplex them. There is no problem. That's our job. We tag every sample with a barcode, and we can multiplex any number of samples. On a NextSeq, we can do about 384 COVID samples at the same time. On MySeq, we can do as much as 400 samples or as low as 1 single sample. So it doesn't matter how many samples you have. Just contact us. We'll help you design your project and we'll help you submit it for sequencing. For the whole genome, small organisms up to 1.2 GB genome size, we can do all of that. The data output is anywhere from 0.5 GB to 100 GB data. For whole exome, we have a 45 MB capture from Shore Select Agilent, and these are whole exome are typically used for identifying genomic variants for cancer panels. Microbiome, Shivaji has already touched up microbiome completely, and I'm not going to go into those details. Metagenome is something that we are able to do on a day-to-day basis. Bioinformatics is something that I'm going to talk about a little more today, because I see that all the talks were more focused on how you can prepare samples and submit them for sequencing. But we are an expert in bioinformatics because we've been doing it for ages. We've developed softwares of our own that are open source. You don't have to buy them. You can just go online and search for the software packages that were developed by us, and you can use them free of cost. Most of our pipelines as well are open source. So that means all pipelines are validly tested. They are, implemented into our process pipeline only after they have passed the validation step. And because they are open source, we are able to update to the latest changes in the algorithms that are used for bioinformatics at a rapid pace. If you are dependent on a proprietary software, you have to buy the license every time a new update comes. So bioinformatics, we can cater to whole genome analysis, microbiome analysis, metagenome analysis, transcriptome, and in silico data mining. That means even if you have done sequencing from a completely different party, you can just submit the data to us and we can do the analysis for you based on your requirements. So the basic idea is we sit with you, discuss about your projects, about your project objectives, whether there's a hypothesis you want to test. We devise a process flow, submit it to you for approval. Once you approve and your questions are set, then we go ahead and ask for the data from you. And you don't have to worry about uploading the data onto a cloud. Because we have in-house servers, we don't upload your data to the cloud, so there is no risk of data security or anything of that sort. Your data is as secure as it is at your place. We retain the data only up to 30 days after the reports are submitted to you, and after that we seek your approval if you want to keep the data for a longer time. Otherwise we destroy the data, but as per NABL and ISO norms, we are supposed to maintain the data for 90 days. So they go on to tape drives where we secure them. There is no access to anybody other than the bioinformatics team and other than the customer. Even the bioinformatics team cannot access your data after 30 days. We have to seek your approval to get the data out. Okay, so unless you lose your data, we don't access it. It's as simple as that. Whole exome we can do basic and advanced, whole genome we can do basic and advanced analysis, whether you have a bacteria, fungi or a virus. There are codes there for every type of analysis, whether you have pangenome analysis, AMR, natural product discovery, or anything of that sort we can do. Microbiome also comprises of components as basic and advanced. Metagenome, we can do shotgun metagenomics or we can do targeted metagenomics, either of them. And transcriptomes, we are able to do prokaryote and eukaryote without any problem. Typically, our process flow goes as we generate a lead. That means you have a query, you approach us, and we then connect. We submit a quote and an order to you. We get the samples picked up from your side. You don't have to worry about how to ship the sample, in what state to ship the sample. Our team will come, they will hand over the material to you, you pack it, you hand it over to our guy, and they will take it. Alright, so it's as simple as that. And then once the sample reaches the head office at Thane, Mumbai, we do processing and then we report to you and then we receive a feedback from you. Our TAT is basically once it reaches the processing site at Thane and till the time we report back the results to you. That's our turnaround time. Typically, it is for NGS. We have the shortest time in the industry. That's about 2 to 3 weeks. Most of the other service providers will take at least about 3 months. For Sanger sequencing, our turnaround time is 3 to 4 days. Alright, so if you have a sample, and if it's a PCR purified product that is just ready for sequencing, you get the results on the 3rd day. Alright, so that's the fastest that we have been able to cater to. I'm not going to cover this slide because Shivaji has already covered this, but I'll walk you through the actual pipeline that we do. This is exome analysis that we do. So we use the SureSelect V8 kit, and once we enrich the targets that we are looking for exonic regions, we run it on a NextSeq 550. So we run it on an NextSeq 550. We generate the FASTQ files which Dr. Karikarte had talked about in the morning. And then we take it through the genetic analysis toolkit, which has the best practices in the industry. So we follow it through this. We generate the VCF files and we then annotate with SIFT, ClinVar, Polyfen, variant effect prediction. All of these are cancer detection, bioalgorithms that are used to identify SNPs or variants that cause different types of cancers or that are associated with clinical evidence to prove that this is the cancer type that's affecting the patient. So all of this is possible, and then we generate an interactive report which we submit to you for actionable insights to better understand the clinical picture. And all of these things are basically ACMG and AMG guideline based. So there is no discrepancy. You can rely on the reports as much as you would rely on the doctor who's prescribing the disease, prescribing the medication. So these are the different types of exome analysis parameters that we deliver. So we actually do a variant classification based on what type of a variant it is. Variant type, SNP, IBS, DEL, SNP class. We do variation, what kind of a variation it is. We do variant classification for each of the parameters that is there. We also do a waterfall plot, which is a population level analysis for the entire population. That if you have a population genetics kind of a study, we do that type of analysis as well. We also have surveillance of emerging viruses. That is one of the key factors that I want to touch. That's because our monkeypox kit was actually developed in a span of 15 days. The first virus was reported. We went to GSAID, we downloaded the sequences, and we downloaded all the monkeypox strains that were available, all smallpox, chickenpox, camelpox, foxpox, dogpox. So all para-variola type of diseases. We've done the bioinformatic analysis and we've come up with a target which is 100% specific only to monkeypox. And that was backed up by our intensive bioinformatic analysis which we were able to cater to. And November 21, Omicron, that's the same thing we did with the COVID. I'm sure everybody who's worked with COVID are aware that there was an S gene dropout, there was an N gene dropout, and there were reports in the news that said, oh, this virus doesn't have the S gene anymore. That's not possible, practically, right? Because otherwise the virus will not be able to infect. So it was just that the kits that were available in the market, they were not binding in the region because that was absent. So the S gene was mutated and the target region was missing. What we did at our end is we actually went back to the genomic sequences of all the new variants. We identified the missing portions and we modified the kits. Now our kits are 100% efficient to detect any type of virus of COVID that can come and cause a disease. All right. So that's the beauty of bioinformatic analysis, and this was done in about 10 business days. Antimicrobial resistance, as I already talked about, the real-time kit that we developed, that was also backed up with pure bioinformatics to support what kind of gene markers are associated with what resistance types. And we have now 6 different panels that cover up to 48 different antibiotics that are of medical relevance. So any organism that's resistant to any of those gene panels will be detected by the real-time kits that we have. Obviously, this also stems from whole genome analysis that you do for a particular organism. So you can do AMR type of profiling, natural product discovery, and all of those things with your gene sequences that you obtain. Microbiome analysis is typically where I started my BSc from. I started with soil, then I moved to glaciers, then I moved to sand dunes, and then I moved to animals. So I have a whole perspective of One Health. We study plants, we study animals, we study pathogens, and today we are doing a study with the KEM Hospital, Pune, where we are actually studying the transition of gut community from a parent, from a mother, to its newborn, and studying it for over a 2-year period. And I'll show you the data in the next couple of slides, so that will be very interesting. So this is the entire assays that we do. There are different types of analysis that we do. Quickly, I'll browse through the link. Hopefully this will work, because that's why I wanted the internet. The internet is fast enough. So while it loads, basically the idea of generating interactive reports is that whenever we submit PDF reports, you are fixed to a framework. You cannot alter, you cannot modify the PDF reports to an extent. But with interactive online reports, we allow the user a freedom to generate the data as much as they want from the sequences that they have. So our algorithms are actually built into the online report. Now, when I say online, it's actually not exposed to the internet. It's a secure link that you only get, and nobody else can see this because we provide you with a password, and that's what the report is all about. So I'll quickly walk through. There's typically a bar that's yet to open. So you have a browsable index or a content on the left panel, and this is the entire report of 16S sequencing. As you can see, you have information about what this method is all about. You have, so yeah, that's the bar that just opened. So you have the entire analysis workflow, like the algorithms that we use. So that helps you understand bioinformatically what we are doing with your data. Not many companies actually reveal what kind of a pipeline they do, but our philosophy is to empower the client. So we share everything that we do with your data. All the quality parameters are then generated. So let's say these are the 10 samples on which we have worked. This is the entire multi-QC report that actually goes to you. You have the fast QC results where you can see the distribution of the sequences in our samples. The samples are prepared by us. So when you prepare samples methodically, you actually get even distribution of all the samples. That's called normalization. If the normalization is not accurate, you will have one sample that will generate 5 lakh sequences, and the other sample will generate only about 10,000 sequences. So there's a disparity, and because of disparity, the sample with low sequence depth is non-usable. So you actually balance it out so that all the samples are at fairly even sequence depth, and that's how you are able to analyze all the samples together. So you have the read quality histograms, you have all parameters are basically included in this entire report. So all of this is available to you. And then you move down to the actual sequence cleaning, where we remove the error sequences from the data set, and we actually do the read pre-processing. These are the algorithms that we use. Tremomatic is very common, where we actually generate, for each step, actually we have a QC step. That means each command that's run on the computer on your data, it is followed by a QC check to ensure that the previous command has run properly. That is to authenticate that the pipeline is following the way it should. So that ensures that the data we deliver to you is accurate, because each step takes its own time to run on the bioinformatics cluster. So we do the final cleaning, and then we generate the data set. We do the error profiling of all your samples. You have the sample inference, read statistics for every sample. You can then assign taxonomy. Now this, when I meant that it is an interactive, you can actually sort the samples by index, by the condition that you are studying, and you can generate different graphs from the same report. All of these graphs are downloadable. You can download each graph at a publication level quality. You don't have to edit them. Okay, so that's the beauty of these interactive reports. You have this heat map that you get for all of your samples. You can generate phylum level distribution, you can generate species level, you can generate genus level distribution from the same data set. So that's how, and then you can have all the indices of alpha diversity. You can have the indices of beta diversity comparisons. These are NMDS plots, PCOA plots. All of these are possible with the same report that you have. And if you have any additional step that you want to do, please let us know. We'll be happy to incorporate it at no additional cost to you. Okay, because for every step that you add, the entire process needs to be redone. So it's better that you tell us upfront that these are the analysis I want, and we'll do it in a single go. So we don't have to repeat the entire process. So I think I'll close this interactive report and move back to my presentation. And this is what I was talking about. Shivaji has already covered this point, like why gut microbiome is very, very important. As you can see, gut microbiome is affected by age, diet, demography, malnutrition, antibiotics and probiotics. And it has an interaction with the human genome and health and disease status. So that's why microbiome has sort of become the key factor in determining most of the dysbiosis states. You have a different community for the gastrointestinal tract, urogenital, skin, oral, nasal. So every portion of your body has a completely different community, and that's why it's important to understand which organism is causing the problem in that particular area. So even the diet has a huge influence. As you can see, in India, we have the prevalence of Prevotella, whereas in Russia, you have all of these 3 different types of organisms which are very different from the Prevotella. And that's because our diet is cooked, Russian diet is purely raw meat. Alright, they just boil in meat. So there's no change in the diversity. And that's what causes a lot of issues. And this is a new field where you have the gut-brain axis. That means your gut and brain are actually communicating to each other, and the community inside your gut is actually changing the type of organisms or the type of behavior you have and the mental state you are in. Okay, so that's the new field that's coming up, and there's a lot of research. And I wanted to briefly touch about the last portion, and that's going to be gut microbiota affects mother's metabolism during the pregnancy. And that's the key factor which we wanted to study. And in this study, basically what we have done is, there's a 7-generation cohort at Vadu Research Station. I'm sure everybody is aware about the Vadu Research Station. And there are 7 generations of subjects that are being followed. There were girls that were about newborns when they were included into the study. Today they are at a marriageable age. Some have already married, some have actually delivered. So all of these 7-generation metadata is available with us and with KEM Hospital, to be precise. And KEM Hospital has collaborated with us, where we have given B12 supplementation to these pregnant ladies, and we have progressed or studied their change in the gut community throughout the delivery process. That means pregnancy, first trimester, second trimester, third trimester, and delivery stage for both the mother and the baby. So this is basically the sampling strategy that we have used. Pre-pregnancy, 28 weeks, delivery, 3 months, 6 months, 9 months, 12 months, 18 months and 24 months. So this is a 700 subject cohort where we are actually doing this entire investigation, and we have just generated some of the preliminary data, which is very, very fascinating. We have studied the stool samples from all the mothers and stool samples from the babies from time of delivery until 2 years. Breast skin swab, because the mothers feed their babies, so the breast skin swabs actually allow us to study the transfer of microbial community from the mother to the babies. Vaginal swabs, because there are studies where they clearly say that C-section deliveries are very different, and regular normal deliveries tend to lead to a change in the community of the newborns. And newborn babies are more healthier when they are born through the normal canal. So that's the difference that we have studied. We have also studied the composition of breast milk as against babies who are bottle fed. So these are different types of objectives that we wanted to study. But this is the data that I'm going to quickly show you. As you can see, if you look at the alpha diversity measure, which is basically just a measure to study how many microorganisms are present in a particular subject. Alright. So if you see, there is a progression in the mother's status from delivery to 12 months, the babies. And post-delivery, there is a shift. Alright. So there is a microbial community which is decreasing in diversity as the mothers change from delivery till 12 months. So the mother's diet has a big influence, and correspondingly, if you will study the community of the newborns, you will see that the community of newborns actually decreases only for the first 3 months, and after first 3 months it sort of stabilizes, and then it starts to mature. This is what happens, and when it matures at about 2 years, at 2 years it stabilizes, where the community stays until the person is adult, and at adult stage it changes again. So that's going to be a major shift that happens. So as I said, it sort of takes a U-turn. It takes a U-turn because for the first 3 months, in most cases, the babies are breastfed by the mother. So as the mother's diversity of the community goes down, the baby's diversity also falls. And after they are moved away from breastfeeding, it tends to take its own shape and it changes. So that's the beauty. And now what we are doing is we are basically able to identify which organisms are changing and how they are changing, what kind of pathways they are doing. All of this is possible because we now have support of bioinformatics. This is pretty much one of the last few slides. Gut microbiota of parents versus babies. So as you can see, these are the 24-year-old mothers and 24-year-old fathers. They have a very high diversity as compared to the babies that are born. So there's a difference, there's a shift. Even though they eat the same food, the transfer of microbial community to the babies is very different. And this is again the same thing where the parents and the babies have a very different shift of the community. We were able to identify the targets which are changing for every type. And as you can see, for the mothers, lactobacillus are very, very prominent. Placebo girls, they have a different community structures. The fathers have a different community structure. And there was a different subgroup of fathers. We were actually able to change or trace it out that they actually had a lower level of nutritional status. So this community of fathers actually were from a poor background, so they had a lower intake of nutritional food. So these are the placebo boys. And last but not the least, I would like to present two main considerations when you are preparing samples for NGS. First of them is what strategy to use. Shivaji has already touched it. Whether you want to use amplicon metagenomics, that means you want to do 16S, 18S, ITS. This will only tell you limited information, as in who's there and what they are capable of, not what they are actually doing. Because what you do here is, based on the 16S sequencing, you identify the type of organism that was present, and from 16S sequencing, you predict, based on the closest representative in the database, what potential functions that organism is able to do. Alright? Now we know that in microbiology, even immunology especially, even subvariants or substrains have a different phenotype, different pathotypes, right? So one substrain is able to cause a disease but the other one is not. So this is just a predictive way of studying. The other way of studying is short-term metagenomics, where you actually study the entire genome of the organism in the environment, and what you are able to do is, you are able to break these entire genomes into smaller pieces, and you sequence each of these smaller pieces, and you sequence them, and you identify functions of each of these portions. And in cases, many cases, if your sequencing depth is good enough, you can actually pretty much circularize the entire genome. These are called metagenome assembled genomes, so MAGs as we call it in our language. So these are MAGs which are able to allow, or which are able to give you identification and information about organisms that are not even culturable in the lab. So even organisms that are not cultured, I am sure everybody who's from microbiology knows that 99% of the microbial community is still unknown, right? We have only been able to culture in the lab 1%, this was about a decade ago. We have moved to about 1.5%, alright? So there's still 98.5% that is still pending to be studied. And these are the organisms that are actually creating a balance onto the environment. Some of the depths of oceans actually have organisms of a single type. So imagine a microbial culture plate inside an ocean in 1 liter of water or 10 liters of water where only a single type of organism is thriving. So that's the beauty of this technology, which has been able to gather this information out from the deeper depths of the oceans. The types of bioinformatic tools that you can use is BioPlug, Mika, PyCrust, Time, Mother, Smash. A lot of different algorithms are there. I've only pointed out those that are open source. So you can go online, use them, try them. If you face any difficulty, feel free to connect with us. We'll be happy to help. Alright? We've developed a lot of our own algorithms which are not listed here, but you can connect with us. This is the typical analysis workflow where you have sample 1, 2, 3, they undergo sequence cleaning, then they undergo data to error correction and profiling. Mind it, most of these analyses are also Windows compatible. So you don't have to necessarily know Linux language or coding to use these packages. That's the beauty of bioinformatics today, because everything is shifting from coding based to GUI based. All of these packages, when I started, they were all R based, command line. I had to really struggle hard to do all this. Today, the newer generation, sir, hum to sirf GUI karenge, humko aur kuch nahi aata. And still we are able to generate the same thing. Because that's the whole beauty. Science should be accessible to everybody. It should not be limited only to the technically sound. And as a medical doctor, I cannot expect you to learn coding, right? So I know that there are limitations, even I have limitations. After 45 years, I've realized that I'm not an expert in sequencing. So that's how we help each other. So it undergoes, data curation, phyloseq, functional profiling, differential abundance, leaf-seq for functional gene abundance, and then finally we report the data to you. And one of the methods that you really need to take care of is the DNA extraction method. When you use in-lab DNA or RNA extraction method, there must be a calculated analysis to see what kind of a community each DNA extraction method is able to capture. We've done our own comparisons, as you can see, with party H, phenol chloroform, and party Q. There is a difference in the median diversity that you are able to harvest. There is a difference in the Shannon index that you are able to harvest. So there's a difference in the community that you harvest. And when you actually look at the level of organisms of each type that are picked out, you will see that one method actually preferentially picks out one organism, whereas the other method doesn't. So there are methods that average isolate from every type of organism, whether it's a gram-positive, gram-negative, UTI or anything. There are other methods that preferentially pick out DNA from gram-negative organisms because they are easy to extract. So these are different types of methods that we use. This is another way of checking that this Prevotella for this one method is extensively picked out, whereas this other method, it is not picked out. So all of these considerations need to be given a deeper thought, and these will help you when you actually do your own experiments. In summary, if you, HiGX 360, which is High Genomics 360, that's the brand name with which we go for genomic solutions. You get an accurate QC from the client. That's a client questionnaire that we get from the client. And you include a sample intake form along with your sample that is submitted to higx360@himedialabs.com. You declare it as a non-biohazardous material because obviously we cannot handle pathogenic material. They have to be inactivated before you send it to us. Packaging is obviously client's responsibility, but pickup and shipping is our responsibility. TAT begins after the first QC report is sent to you. After HM code generation, we actually do the Sanger sequencing and report is sent to the client by the technical team. The beauty of IGX 360 is that you get to interact with me directly or my bioinformatician directly. So you don't have a middleman ki mujhe isse baat karni hai, usse baat karni hai. You can directly dial my number and we'll be happy to help. Costing is obviously very affordable because that's the primary factor. Sequencing for Sanger starts anywhere from 300 rupees. And for NGS starts anywhere from 3,000 rupees. Okay, so that's the beauty. All my research and studies over the past years, the data that I have shared with you today are funded by all of these organizations. I'm very thankful to USDA Agricultural Research Foundation and NSF National Science Foundation USA who have actually funded some of this work. And you can write to us at higx360@himedialabs.com. And thank you, I'll be happy to take any questions.
One thing you have like asked, there is a disease called as a hand, foot and mouth disease. Anything to relate to the monkeypox? Have you worked on that?
Yes, we are actually working on it. It's internal use, but I can share. We are working on a kit that's coming out soon. It will be a real-time PCR-based detection method that will segregate monkeypox, FMD, and the other boxes.
Even the box is caused by the toxicity virus.
Yes.
Totally different than the other box.
Exactly.
That's why I'm interested.
Yeah. Hopefully in about a month you will have the kit.
I will request you for that.
Yes, please.
Hello, sir.
Yes.
Tuberculosis.
Tuberculosis, we already have kits, real-time PCR based kits, but if you are interested in NGS, we are already working with D.Y. Patil Hospital, Mumbai. We are doing a study where TB treatment, post-treatment and how it actually modulates the microbial community, that's also we are working. So there are a lot of projects that are in pipeline and we are doing all kind of studies where we can support. Yeah, I didn't mention because there was, not enough time to cover everything. Yeah.
Well done.
Thank you so much.
Very interactive speech, Dr. Kamlesh sir. I request Dr. Smriti to please give his memento and a certificate to sir.
We are privileged to operate this. He has been very informative and very educative.
Yes, sir. Please, sir.
Take care.
I now welcome Dr. Vinay Verghese. He's a PhD scholar from Manipal University. He's currently working with Thermo Scientific.